Hello guys, and today we are going to discuss about the principal cells of the late distal tubules and the cortical collecting tubules. Principal cells are special type of cells that are present in the late distal tubule as well as the collecting tubules, or the cortical collecting tubules to be more specific. Along with the principal cells, other type of cells are intercalated cells. We have discussed in the previous lectures about the function and structures and properties of the intercalated cells. So principal cells, talking about the structure and the transporters. um, on the luminal area there are two type of transporters in the principal cells. First is the sodium transporters and the second is the potassium transporters. And on the basolateral surface or the membrane there are sodium potassium ATPases. And like most of the cells of the nephron, the distal and the cortical collecting tubule principal cells also have sodium potassium ATPases. What is the function of sodium potassium ATPase? They pump sodium out and potassium into the cell. So as a result, the sodium concentration inside the cell decreases and the potassium concentration inside the cell increases. And on the luminal area, this potassium is thrown out of the cell along the concentration gradient, and the sodium coming in the tubular fluid in front of the principal cell is reabsorbed into the cell along the concentration gradient of the sodium, because the sodium inside the tubular fluid is high and potassium in the tubular fluid is low. So as the along the concentration gradient, these ions will going to move. And the most important thing, I want you to know that effect of aldosterone on the principal cells. First of all, what is aldosterone? Aldosterone is a hormone that is produced by the zona glomerulosa of the adrenal cortex. This is the zona glomerulosa of the adrenal cortex. Adrenal cortex is the suprarenal gland part. It has two parts, adrenal cortex and adrenal medulla. This aldosterone is produced by one of the layers of the adrenal cortex, which is zona glomerulosa. The other two are zona fasciculata and third is the zona reticularis. So aldosterone hormone has receptors that is present inside the principal cells. Let me show you how the receptor of the aldosterone will going to function in the principal cell. So first of all, this is the receptor. The green, this red one is the receptor for the aldosterone, and this is the hormone itself, aldosterone. When aldosterone binds to the receptor inside the cell, this hormone receptor complex, aldosterone plus receptor complex, will translocate to the nucleus of the principal cell. This is the nucleus. And inside the nucleus, there are multiple genes. What kind of genes are present inside the nucleus? This one, the gene for the potassium channel, the gene for the sodium channel, and the gene for the sodium potassium ATPase. So when the aldosterone receptor complex translocates to the nucleus of the principal cell, it activates the genes for sodium channels and the potassium channels on the luminal membrane and the sodium potassium ATPase on the basolateral membrane. What does it mean? It means that these genes are going to undergo the process of central dogma. It will form messenger RNA via the process of transcription, and messenger RNA will form proteins that are channels by the process of translation. What does it mean? It means that the number of sodium potassium ATPase will increase on the basolateral membrane, the number of potassium channel will increase on the luminal membrane, and the number of sodium channel will increase on the luminal membrane. It means that under the influence of aldosterone, sodium will be pulled out of the cell into the interstitium, potassium will be taken in. which means that more potassium loss and more sodium reabsorption will occur along the concentration gradient. It means that under the influence of aldosterone, there will be salt and water retention inside the principal cells. Okay, there will be salt and water retention, and potassium will be excreted, which means kaliuresis. Kaliuresis means potassium in the urine. And other conditions in which the aldosterone level increases in the body, for example, congestive heart failure, hypovolemic shock, and Conn syndrome. Conn syndrome is a tumor in the zona glomerulosa in which all these conditions result in high level of aldosterone in our bloodstream, which means that this aldosterone eventually comes to the principal cell, and the aldosterone receptor complex will activate the genes that are responsible for the synthesis of these proteins, and there will be increase in the salt and water retention and potassium excretion. Now you know that why under the influence of aldosterone there is sodium and water reabsorption and potassium excretion, that is kaliuresis. This part, which means the late distal tubule and the cortical collecting tubules, are responsible for the reabsorption of 3 to 7% of sodium, to be more specific, because 65% of sodium has already been reabsorbed in the PCT and 25% has been reabsorbed in the thick part of ascending limb of loop of Henle, and approximately 5 to 6% has been reabsorbed in the early distal tubule. So what is remaining? Maybe 3 to 7%. So this part is dealing with that 3 to 7% of sodium, which means that fine tuning of sodium under the influence of aldosterone will be occurring in the late distal tubule and the cortical collecting tubule. There are two types of diuretics. Diuretics means that those drugs that produces or promote the diuresis process. Okay, there are two types. First is the potassium losing diuretics, and the second is the potassium sparing diuretics. Okay, let's talk about the potassium losing diuretics. It means that they will promote more potassium excretion in the urine. Thiazide diuretics and the loop diuretics are both potassium losing diuretics. It means that, as you remember from the previous lecture, this is the thick part of the ascending limb of loop of Henle. Okay, and on in the thick part of ascending limb of loop of Henle, there are specialized cells that has special transporters known as the sodium potassium 2 chloride co-transporter. They have sodium potassium 2 chloride co-transporter on the luminal membrane. And in the early distal tubule, this is the early distal tubule. Okay, there are special cells that have special type of transporter on the luminal membrane known as the sodium chloride co-transporter. So these two diuretics, the loop diuretics and the thiazide diuretic, will work on these transporters. What they will going to do? They will inhibit these transporters. They will inhibit these transporters. When the loop diuretic inhibit the sodium potassium 2 chloride co-transporter, can the sodium be reabsorbed? No. Can the potassium be reabsorbed? No. And can the chloride be reabsorbed? It cannot. So all these solutes will move down the drain. They will move forward. And the thiazide diuretic will block the sodium chloride co-transporter. Can the sodium be moved in? No, sodium cannot be reabsorbed. And chloride, no, chloride can also not be reabsorbed. So they will also move forward. What does it mean? It means that these diuretics will bring extra load of cations, which are sodium, anions, which are chloride and bicarbs, and water in front of the principal cells, because principal cells are present in the late distal tubule, right? Principal cells are present somewhat here and here. So it means that under the influence of the loop diuretics and the thiazide diuretics, they will bring extra load of cations and anions in the lumen in front of the principal cell tubules. So potassium losing diuretics, how does this work? Be concentrated that first, number one is that the solutes are present, more amount of solutes are present in the tubular fluid. So amount of water will also be increased because water will be attracted towards the solutes. So water concentration inside the tubular fluid is high. So is the water flow is high also? Yes, the water flow in the tubule is high. What does it mean? It means that this water will wash away potassium from the principal cells into the urine, kaliuresis. Second, the extra amount of cations like extra amount of anion, sorry, the chloride ion which is negative, the bicarbonate which is negative, they will attract the potassium and they will cause its excretion. Again, kaliuresis. The third thing is that the extra amount of sodium will come here. Okay, the extra amount of sodium will come here because under the influence of loop diuretics and thiazide diuretics, the sodium has not been reabsorbed. So where does the sodium go? Of course, in the tubular fluid, forward in the tubular fluid. So extra amount of sodium is present in front of the principal cell. So it will cause the sodium reabsorption along the concentration gradient. As you know that on the principal cell basolateral membrane, there are sodium potassium ATPases. They are constantly throwing the sodium out. Concentration of sodium inside the principal cell is decreased and concentration of sodium in the lumen is increased due to the effect of thiazide and loop diuretics. Sodium will move into the cell and it will be thrown out of the cell by the sodium potassium ATPase. It means that more sodium will be thrown out of the cell, more potassium will be bring into the cell, and this potassium is lost into the urine. It means that kaliuresis. Under these three mechanisms, these diuretics will promote the potassium loss into the urine and they will produce kaliuresis. Eventually, they will cause a decrease in the blood potassium level, which means hypokalemia. Okay, they will cause hypokalemia. Okay, because in the blood the potassium concentration has been decreased. The other type of diuretics are the potassium sparing diuretics. What does it mean? Potassium sparing diuretics are those that will prevent the potassium loss into the urine. And these diuretics, in contrast to the potassium losing diuretics, work on the principal cells. They will directly work on the principal cell. So let's discuss how does the potassium sparing diuretics are working. The first example is spironolactone. Okay, this one is the spironolactone. Spironolactone will inhibit the receptor for the aldosterone. It will inhibit the receptor for the aldosterone. When this receptor is inhibited, can aldosterone bind to the receptor? No, it cannot. It means that genes cannot be activated. When genes cannot be activated, the synthesis of sodium channels, synthesis of potassium channels, and synthesis of the sodium potassium ATPase is decreased. So it means the sodium cannot be reabsorbed. Sodium cannot be thrown out of the cell by the sodium potassium ATPase, and potassium cannot be taken in by sodium potassium ATPase. So less potassium is lost into the urine by the potassium channels. It means that spironolactone, i.e. the potassium sparing diuretic, will prevent the potassium loss into the urine indirectly. Okay. The second type of potassium sparing diuretic is the triamterene and amiloride. Triamterene and amiloride will directly inhibit the sodium channels, these luminal sodium reabsorbing channels. When they are inhibited or they are dysfunctioned, can sodium be reabsorbed? No. Can sodium be thrown out of the cell? No. Can potassium be brought into the cell? No. So can potassium move out of the cell? No. It means that these triamterene and amiloride will directly inhibit the potassium lost into the urine by inhibiting the sodium channel in the luminal membrane. Our last discussion is about the effect of antidiuretic hormone on the principal cells, and it is very important because antidiuretic hormone is antidiuretic hormone. It means that it will not promote diuresis. So how does it work? This is a special principal cell, and it has on the basolateral membrane it has aquaporin 3 and aquaporin 4. Aquaporins are special water channels that are present in specific cells, and it is embedded in the membrane, more specifically the basolateral membrane in case of the principal cells. Number 3 and number 4. And aquaporin 2 is also present inside the cytoplasm, but it is like joined with the vesicles. It is planted in the vesicles. So first of all, in the absence of ADH, when there is no ADH, no water channel is present in the luminal membrane. So again, let me draw a nephron for you. This is a nephron, this is the PCT, the proximal convoluted tubule, the descending limb, okay, and then the thin ascending limb, the thick ascending limb, the early distal, late distal, cortical collecting tubule, and the medullary collecting tubules. Okay, so from the PCT and along the descending limb, this part is under all physiological circumstances is permeable to water. Okay, it's very important to know that this area of nephron, the proximal convoluted tubule, the thin descending limb, and some part of the loop of Henle, right? It is permeable to water under all circumstances. But when the story changes, the thin ascending limb, the thick ascending limb, and the early distal tubule. These parts of nephron are under all physiological circumstances impermeable to water but permeable to solutes. The last part of the nephron, which is the late distal tubule, the cortical collecting tubule, and the medullary collecting tubules. What do you say? Either they are permeable to water or not. I will put a question mark on these tubules because the water reabsorption in this part of nephron is dependent on the antidiuretic hormone. Under the influence of antidiuretic hormone, it will cause the water reabsorption, and in the absence of ADH, there will be no water reabsorption. So first thing is first, in the absence of ADH, there are no water channels present on the luminal membrane. Okay, there are no water channels. The membrane is absolutely impermeable to water in the absence of the ADH. But in the presence of ADH, the ADH will bind to the receptor on the basolateral membrane or the membrane of the principal cell. The receptor is a 7-passed receptor, or it is also known as the serpentine receptor. This receptor will stimulate the G stimulatory protein. This G stimulatory protein will in turn stimulate the adenylyl cyclase. Adenylyl cyclase will convert ATP to CAMP, cyclic adenosine monophosphate. This CAMP will upregulate the function of protein kinase A, and this protein kinase A has the function of phosphorylating some proteins. What will it phosphorylate? It will cause the phosphorylation of certain transporters that will cause the movement of vesicle towards the luminal membrane, and when this vesicle is moving towards the luminal membrane, eventually this vesicle membrane will merge with the luminal membrane, and these aquaporin 2 are expressed or planted on the luminal membrane. It means that under the influence of ADH, the luminal membrane starts to become water permeable, and water will be pulled from the lumen into the cell and from the cell into the hyperosmolar interstitium. As you know that the interstitium in the medulla of the nephron is highly hyperosmotic. The osmotical gradient is increasing from cortex to the medulla of the kidney. So it will make the urine concentrated because a lot of water has been reabsorbed from the final part of the urine, and urine will be concentrated with the solutes because water has been reabsorbed from that. It will make the urine more concentrated. Let's talk about the final part of the nephron, that is the medullary collecting tubule. We have discussed in detail about the proximal convoluted tubule. the descending limb of loop of Henle, the thin ascending limb of loop of Henle, the thick ascending limb of loop of Henle. Then we have talked in detail about the proximal, the early distal tubules. Okay, this is the early distal tubule, this one, and this is the late distal tubule. And we have talked in detail about the cortical collecting tubule, and now we are talking about the medullary collecting tubules. Medullary collecting tubules has some cells that function in somewhat same manner as compared to the early distal tubule, the late distal tubule, and the cortical collecting tubules. For example, in the presence of ADH, these cells become water permeable. It means that this cell has the function similar to the principal cell. And the cells of the medullary collecting tubules can also actively secrete protons into the lumen and bicarbonates into the interstitium, and that bicarbonate can be reabsorbed into the blood. So this function is similar to what? Intercalated cells. Okay, and this part is also concerned with water reabsorption, potassium secretion, and sodium reabsorption. So it has a mixture of similar functions as compared to other tubules, other tubular cells. But the medullary collecting tubules has special cells for urea reabsorption. It means that when the tubular fluid reaches the medullary collecting tubule, okay, let me draw a nephron again. This is the Bowman's capsule, the proximal convoluted tubule, the thin descending limb. Okay, this is a loop of Henle, the thin ascending limb of loop of Henle. Then comes the thick ascending limb, the early distal tubule, late distal tubule, the cortical collecting tubule, and the finally the medullary collecting tubules. Some amount of urea is also reabsorbed in the proximal convoluted tubule, but this part of the nephron is highly impermeable to urea reabsorption. So, when the tubular fluid reaches the medullary collecting tubule, it is highly concentrated with the urea. There are special transporters in the medullary collecting tubules that will cause the urea reabsorption, and this urea will be moved out into the medullary interstitium, and this urea, the urea present in the medullary interstitium, will be taken up by the loop of Henle. Loop of Henle is permeable to urea, and this urea will again become the part of the tubular fluid, and the cycle moves on. Of course, some part of urea will be lost into the urine, but before losing into the urine, this urea will be reabsorbed into the medullary interstitium, taken up by the loop of Henle, and recycled into the tubular fluid. This is known as the urea recycling. It is known as the urea recycling as nephron is concerned. Okay, this is the urea recycling process in the nephron tubules. Medullary collecting tubules have some specific functions for urea handling. Okay, so this was for today, and in the next lecture we'll discuss further about the urine concentration and dilution processes, and I will see you in the next video. Till then, goodbye.